it is sensitive to water diffusion in tissue. With diffusion MRI, we can reconstruct brain connections with fiber tracking and wet matter tract identification. We can also reconstruct tissue microstructure with some well-designed biophysical models. Also, for some brain lesions, they manifest on diffusion-weighted images. So that's another application of diffusion MRI. So today we are most interested in white matter tract segmentation. So we will talk about how to obtain brain connections with fiber tracking and then segment these white matter tracts. The acquisition of diffusion MRI is actually more complicated than other structural MRI images like T1 weighted images, T2 weighted images. So given a brain, we need to apply a number of different diffusion gradients, which gives us a number of different 3D MRI images. On the other hand, we can design some signal model that relates the tissue organization of interest to the signals we have observed. Then with some model fitting, we can infer the tissue information. In today's presentation, we're interested in the directional information of tissue so that we can infer how the fiber tracts propagate in the brain. The diffusion tensor imaging model is probably the simplest model for such inference of tissue organization. Here is a mathematical formulation. Here S0 is some diffusion-free signal where we do not apply any diffusion gradient, and B value is some parameter we determine during the imaging process. G represents the diffusion gradient direction, and D is the unknown tensor we want to use to describe the diffusion process. It is a positive semi-definite matrix, so it has actually 6 independent unknown variables. Based on the signals we observe associated with each diffusion gradient, we can estimate this diffusion tensor. A simple or straightforward way is to convert this equation into a linear form with logarithm transformation. Then with a bunch of different observed y's, we can use, for example, least square estimation to estimate these unknown variables in the diffusion tensor. So after we have the diffusion tensor, we can have some information about the diffusion process. Because the diffusion actually follows the direction of the axons, we can use its principal eigenvector to represent the fiber orientation. So that's where the axons, that's the direction where the axons propagates. Or here's a graphical illustration. We can use the eigenvectors and eigenvalues of the diffusion tensor and use a ellipsoid to represent this diffusion tensor. This is the primary eigenvector here. So this direction represents the diffusion direction, and in this way, we have the fiber orientation. Okay. However, the diffusion tensor is limited in that it only can represent one fiber orientation. But in real life, our brain has crossing fibers, meaning that we can have different fiber tracts cross with each other. So in one voxel, we could have multiple fiber orientations. To address this problem, we can use a different modeling. For example, here we have this very classic spherical deconvolution strategy. Suppose in this one voxel, we can have 2 different fiber orientations represented by these 2 blue sticks. The green surfaces represent the diffusion signals associated with these 2 directions. Along this direction, the diffusion signal is lowest because it is mostly attenuated. And orthogonal to this direction, we have larger signals. Then we can linearly combine these different signals and have a model of the signal in this voxel with crossing fibers. So based on the observed signals, we can try to do a model fitting to estimate what these two directions are. There are a few things we need to determine. For example, the response function. Given a fiber orientation here, how do we model the signal associated with different diffusion directions? Also, the estimation problem is very ill-posed. We need to apply some constraint for more stable estimation or optimization. Also, how do we represent these different fiber orientations and what method we use to solve the inverse problem? There can be multiple different choices. One classic way is to do this with constrained spherical deconvolution, where we have this least square problem, regularized least squares problem, where we first minimize the difference between our modeling and observed signal, as well as some regularization on the fiber orientations. Since I'm told this is an educational presentation, I will not go into the deep technical details, but if you're interested, you can look at the papers on constrained spherical deconvolution, as well as its more advanced versions. Okay. Now that we have the fiber orientations, we can perform tractography or fiber tracking. It refers to the reconstruction of fiber tracks as 3D streamlines. So here's a graphical illustration. Given all these directional information, we can do this fiber tracking, which gives us all these different 3D streamlines that represents our fiber tracks, white matter tracks in the human brain. So if we look at this toy example here, we have 4 different voxels. What we do is we start from some seed voxel, for example, here. Then, based on the fiber orientations calculated from the tensors or other representations, we propagate from the seed voxel. For example, if we do a nearest neighbor interpolation, we propagate this streamline until we reach the edge of this voxel. Then in the next voxel, the streamline is propagated according to the fiber orientations here. Similarly, if we start from this voxel, we do the propagation here until we reach the boundary here. Then we do the next propagation according to this direction. So this is how, in principle, we can do the fiber tracking process. Of course, there can be some variants for smoother fiber tracking results. We can do interpolation of fiber orientations along this trajectory. Also, we need to determine when we want to stop the tracking process, because our prior knowledge tells us that the axons start from some gray matter voxels, and it also terminates at different gray matter voxels. So the first termination criteria is the fractional anisotropy. Briefly, fractional anisotropy is computed from the diffusion tensor. It is greater when we are in white matter, and it is lower when we are in gray matter. So when the FA, the fractional anisotropy, decreases below some threshold, usually we just terminate our propagation process. Also, generally, we believe that our axons do not have a big turning angle. Okay, it is a very smooth, it is generally a smooth structure. So if the turning angle of our propagation is very large, we tend to terminate the fiber tracking process. So here's some more graphical examples. For example, suppose we start from here. We just follow the direction of the diffusion tensor and do this fiber tracking step by step. So we have a bunch of different points along this streamline. So these streamlines are represented with these ordered points in the 3D space. Okay, suppose we have some whole brain tractography results. Then we are interested in white matter tract segmentation. The white matter tracts refer to a bundle of anatomically defined fiber tracts. Different white matter tracts may be involved in different functions and diseases. So we are either in scientific studies or in clinical studies, we're interested in specific white matter tracts. So what we can do is to do a streamlined parcellation based on the whole brain tractography. This is an example. We can, starting from this whole brain tractography, we can label these streamlines and have different groups of white matter bundles. Okay, each of them represent a different fiber bundle, and they could be associated with different functions and diseases. So white matter tract segmentation in this way identifies specific white matter tracts and brings deeper insights into brain development, function, and diseases. It also provides a quantitative tool for the analysis of brain characteristics. So how do we do this streamlined parcellation? We can do this either manually or automatically. For example, suppose we have the diffusion scan. We first do a whole brain tractography. Then, based on our prior anatomical knowledge, we could have some idea about, for a specific tract, we have some ideas about where it should go through. Okay, so we can draw some inclusive RIs to select the streamlines we are interested in. Also, this result could include a lot of false positives. Okay, we also know where the fiber tract should not go to. So we can also draw some exclusive RIs to further remove undesired fiber streamlines. In the end, we can look at this result, and probably we are not happy with the result. We need to do some further refinement, or we want to just refine our inclusive or exclusive RIs to get a better streamline parcellation result. So we can do this repeatedly with refinement of these RIs. So this is how we can do this manually. Also, we can do this automatically by replacing the manual region of interest with some atlas, registered atlas. And this final manual refinement can be replaced by some machine learning streamline selection techniques. In this way, with this pipeline, we can do the streamline parcellation either manually or automatically. And finally, from the whole brain tractography result, we can get a specific tract. Here's actually another way of doing the white matter tract segmentation, which is volumetric segmentation. If you're familiar with image segmentation, this concept is very similar to the conventional standard image segmentation. What we can do is to directly label the voxels like a standard image segmentation problem. We don't need to do the tractography. Instead, we are given the input of diffusion scans or fiber annotation maps or diffusion tensor maps. We use some segmentation algorithm like atlas registration, Markov random field, random forest, etc., and directly produce the tract label map. Okay, and compared with the streamline parcellation, it has both advantages and disadvantages. For example, it does not require fiber tracking, but also the downside is that this volumetric segmentation cannot give connectivity strength information, like how many streamlines are connecting from region 1 to region 2, etc. All right, since we want to talk about deep learning-based white matter tract segmentation, we also will introduce some basic knowledge about deep learning. So deep learning refers to artificial neural networks nowadays, which are inspired by brain or biological neural networks that is very well organized to process information. The basic mechanism is the connections of a large number of structural simple elements So artificial neural networks are also motivated in this way to mimic this simple and elegant mechanism. And the simplest element in artificial neural networks is the perceptron. Okay, here is the mathematical formulation. We have some input vector v here. We have some parameters in the perceptron, parameters w here. The perceptron just does a linear transformation of the input vector followed by some nonlinear function. However, the perceptron itself is very simple and cannot have a very good discriminative capability. So what we do is we can concatenate multiple perceptrons and which gives us the multi-layer neural network. The concept is very simple. We have one perceptron here. Its output is fed into another perceptron. In this way, we have more representative power of the neural network. The parameters are still unknown in the neural network, but we can just learn these parameters. We can formulate some error function for minimization. For example, for classification, we use cross entropy. And because most of the time the error function is nonlinear and non-convex, there's no analytic solution. So what we do is we use gradient descent to find the optimal parameters. Since we have multiple layers, usually we do this optimization with backpropagation for efficiency. And for the details, we're not going through the details of these optimization techniques, but you can find that on Wikipedia or other materials. The problem is actually we are dealing with images. So the multiple layer perceptrons are not quite suitable for processing images. Because for images, multiple layer perceptrons takes vectors as input. If we do the vectorization of the image, first it destroys the structural and configuration information in the images. Also, it means that we will have a huge number of unknown parameters in the multiple layer perceptron. So people have designed convolutional neural networks to process images. What we have is a bunch of unknown convolutional kernels. With each kernel, we can process this image and get a feature map. With a number of different kernels, we get a number of different feature maps. And we do this convolution sequentially with different layers. And also, we can do some subsampling during the process. Finally, with some global averaging or global pooling, we convert the feature map into a feature vector of a fixed length. Then we can use these multiple layer perceptrons to further process these features. Finally, it gives us a classification result. So this is a typical structure for CNN-based classification network. Also, we are interested in segmentation. What we want to produce is a map result, okay, not a single classification result. In this case, we can use other network architectures. A very classic one is the UNet structure we list here. We still have these convolutional layers. We have downsampling layers, but we also have these upsampling layers and followed by other convolutional operations. In the end, the output has the same size of the input. Also, for the UNet structure, we have these skip connections that just feed these features directly to this part. So anyway, with this network structure, we can have a output size that is different, that is the same as the input size. Also, we have other more advanced network architectures like transformers, but since in wet matrix track segmentation, the transformers have not been quite applied yet, so we'll skip the details. Briefly, transformers allows us to model long-range dependencies, okay? Because for the convolutional operations, it only uses local information to obtain these feature maps, which may neglect the non-local long-range interactions. So the transformers use this so-called self-attention so that you can have interaction between voxels that are very far away from each other. So that's the basic idea. Now that we have the basic knowledge of tract segmentation and deep learning, I will show some examples of how to apply deep learning techniques for wet matter tract segmentation. We start with a streamline parcellation example. So this is the deep WMA algorithm. It has created a large-scale training tractography dataset of 1 million labeled fibers from this atlas. And it also has developed a novel fiber feature descriptor called fiber map. Then it used this fiber map to do the classification based with the help of a convolutional neural network. So let me briefly go through the process. This is the atlas for training. In the atlas, we not only have the streamlines, we also have the labels of each streamline. For example, here we have the cortical spinal tract fibers. We have the arcuate fasciculus fibers. Also, there are some fibers that do not have an anatomically defined label. They are all grouped into these false positive fibers. Since the streamlines are represented by ordered points along the streamline, they can have different numbers of points. So we need to find a unified representation for different streamlines. In this work, they use a so-called fiber map, which converts the original points here into multiple images. Then we can feed these images into some classification CNN model, which gives us the label, the classification result of this streamline. So based on the atlas, we can use all the 1 million fiber streamlines to train this CNN model. Then with the trained CNN model, given some new tractography results, given this unlabeled tractography results, we can label each streamline in this whole brain tractography result with the trained CNN here, which gives us the predicted streamline parcellation result. And this work has also been validated extensively. Here, I'm just showing one graphical example. It has been compared with other techniques and achieved more accurate parcellation accuracy. Another example is how to use deep learning to do the volumetric segmentation. As we have mentioned, we can use a UNet structure for the segmentation. Here are two similar works for the same task with slight differences. Since TrackSec is more widely used in scientific studies, I'll use TrackSec as an example here. It takes the fiber orientation maps computed from the diffusion scan as input. Also, it actually uses 2D UNets, but our fiber orientation map is 3D. So it has 3 different views for processing the inputs. We have chronal slices, axial slices, and sagittal slices. They are all fed into the 2D UNets. And in the end, we will have 3 different segmentation results. And they are fed into the second convolutional neural network to average all these different results to give the final volumetric segmentation maps. Actually, in practice, we most of the time we just use the first UNet and directly average the results of the first UNet. So in TrackSec, they have labeled 72 major wet matter tracts in the brain, and they are graphically shown here. Also, our group has improved this TrackSec strategy with some other ideas. Because for some tracks, like the fornix or anterior commutator, they are very thin. And for the uncinate fasciculus, they have very curved structure. The segmentation of these tracts can still be challenging with the tract-seg algorithm. We try to improve the segmentation quality for these challenging tracts. And to do that, we want to incorporate some characteristics of white matter tracts. So we know that these tracts may overlap or cross. It means that in the voxel, multiple tracts can coexist. It also means that different white matter tracts can be correlated. So in this way, we incorporate this correlation with the label embedding. This means that we can map the tract label vector into a lower dimension embedding space because they have correlations. So this is what our network with label embedding looks like. Instead of directly predicting the tract labels, we predict a lower dimensional embedded vectors, which reduces the difficulty in the estimation. Then we have another reconstruction module that just recovers the label maps from these lower dimensional embedded vectors. Okay. This is the general idea. Again, this is educational talk, and I will not go into the details of this one. If you're interested, you may refer to our paper published 2 years ago. And we have evaluated the result both qualitatively and quantitatively. I'll just briefly show this example here. Compared with these conventional results, for the segmentation of the fornix, we are able to preserve its continuous topology here. We have also performed the quantitative evaluation by computing the dice coefficient, where we have achieved improved dice scores. Okay, at the end of this presentation, I will show some public resources and also summarize our talk today. There are actually many useful public resources on the website, on the internet. I'm only listing 3 examples that I have been using a lot. There are also other different very valuable resources. Here I'm listing the MRTrix, the FSL, and DiPi. For both MRTrix and FSL, they have provided graphical user interface, and all 3 of them have provided APIs that we can call to develop our own algorithms. Okay, and they're very classic software for processing diffusion MRI, and they actually have more functionalities than processing diffusion MRI. All right, to briefly summarize, today we have introduced the basics of diffusion MRI, like the acquisition, like how to estimate fiber orientations, then based on the fiber orientations, how to do the tractography or fiber tracking. Given the fiber tracking results, we can parcellate them into different white matter tracks. We have also introduced the basic concepts of in deep learning, like perceptron, multi-layer perceptron, CNN transformer. Then we have given 2 examples about how to apply deep learning to white matter tract segmentation. There may also be some future works on white matter tract segmentation. For example, how to apply them reliably to real-life data is still a challenge. In real life, data can originate from different hospitals. We have really very, very heterogeneous clinical data. They are scanned from different vendors. They're scanned on scanners with different vendors using different protocols. They can have different SNRs. So currently, there can still be failures when we have some new data from an unknown hospital. Also, what may be interesting is that, for example, in clinical practice, we sometimes, or actually most of the time in China, we do not acquire diffusion MRI. What we have is structural images like T1 or T2 weighted images. So how is it possible to perform tract segmentation on these structural images? That's also something that's worth exploring. There are already some pioneer works on exploring the topic, and we are going to do that as well. Okay, that's what we want to introduce today. Also, special thanks to Professor Fan Zhang for providing valuable slides. And thank you for listening. I'll be happy to answer questions. Thank you.
Thank you very much for this, for your talk, and a lot of very useful information for people that are not familiar with machine learning. So thank you for